Good afternoon. Thank you for coming. It's always fun for me. I feel like I'm planning a party and I'm not sure who's going to show. So I really appreciate your time, especially in light of the fact that it's 80 degrees and that maybe everyone went golfing. So thank you so much. I'm going to let Dr. Peter Boling introduce himself. He's quite credentialed, but this is, I think, the highlight of our Active Aging Week. And what I hope this does is propel momentum forward to the walk to end Alzheimer's. November 4th, you heard from Amy yesterday, she would really like to have 100 bodies walking with the Cedar Field shirts on. They actually came in today and they're very sharp. So think about that as you're seated. We celebrated the fundraising with a large check signing yesterday, but just think that maybe this spurs you a little bit more to the yes, I'd love to participate. Thank you, and here's Dr. Boling.
Hello, everybody. So, I'm Peter Boling. I've been here in Richmond since June of 1981, when I was an intern at what we called the Medical College of Virginia, now VCU Health System. And I did my training down there, then stayed on as faculty. So I've been on the faculty there for 39 and a half, almost 39 and a half years, which is, quite a while. And over the course of that time, I started out not knowing what I was going to do with myself, doing some work in the clinic, and I was given the opportunity to start making house calls. So that was my launch into geriatrics. I had no intention of being a geriatrician at the beginning of my course in training, but then I found myself more and more realizing that this is where I belong. So since that time, my career path has been all around geriatrics in one way or another, a lot of it in home health. But of course, in geriatrics, one of the things that we have to deal with on a regular basis is impairment in cognition, right? So mind's not working as well as it used to, or it doesn't work like it used to, and then people start to worry about it. Sometimes it becomes a real problem. So what I'm going to do today, and I'm hoping that you're ready for a ride through science, because I brought my science pack with me. I'm going to talk to you in terms that I think will make the science understandable, but in order to really appreciate the complexity of this subject, you do have to spend some time in the place where I dwell, which is in the world of medical science and applied medical science. So I'm a doctor, right? But I also am a professor at VCU. I'm the chairman of now the chief of the Division of Geriatric Medicine, position I've had for about 10 years. And I have been out here to Cedar Field a few times over the course of my career to see a few patients. So I'm really glad to be back here again and to see your smiling faces. If I see people start falling out of their chairs, I'm going to recognize that that's a clue that I probably should freshen it up or something else. Maybe we'll do some jumping jacks or something along those lines. I don't know. So, okay, if you have questions, please raise your hand and I will answer your questions as best I can. I'll have some time right at the end. We can go back over anything you need me to touch on. Everybody ready? We're going science. Okay. I got the clicker. He said, don't touch these two buttons, whatever you do. These are the questions that I put up there, right? So how do patients, families, and healthcare providers distinguish between senior moments? Who's had a senior moment? I certainly have. Mild cognitive impairment and dementia, so that, it's not all the same, right? We have senior moments even when we don't have really anything seriously wrong with our cognitive function. How much can we gain from the available prescription medicines and over-the-counter supplements for memory? I'm going to try to answer that question for you. Another question that I get quite a bit is, if members of my family have dementia, is it likely that I'm also going to be affected? Then what preventive strategies can we employ to reduce the chance of getting dementia, right? And then finally, I'm going to finish up with some new stuff that's really pretty exciting that's just come onto the scene in the last 2 years after many years of research and build-up. And that's really the big news in dementia care at the moment. But it's not going I'm going to tell you, sort of spoiler alert, it's not going to solve all of our problems right out of the gate. Okay, so there. All right, so it's always necessary with a talk like this to start off reminding everybody that this is a big problem. It's a big problem. You already know it. You may have some family members that have this condition. Some of you may be in some way affected. You certainly have some awareness of it, and probably a little bit of fear and anxiety around the possibility of developing this terrible condition. It's also super expensive, as you can see here, and increasingly so. And a good part of the cost is actually not to the patient and to the Medicare program, which is paying, the bills. But a lot of the cost is stuff we don't see. It's the burden that falls on members of the family and the others that are providing support for the individuals. Okay, so now I'm just going to take you through what our normal process looks like when somebody comes to me and says, and oftentimes the person saying this to me is a daughter or a son, that we're concerned about this individual, seems not to be processing information as well as they used to. So what I teach all my students is the first thing we need to do is we need to understand what's normal. What's a normal circumstance when somebody's getting older? Because everybody knows in this room, I can see from looking around, everybody is aware things have changed some from when you were 20. And yeah? Right. So I'm very much aware of that myself. On Wednesday of next week, my right hip is going to go into the bucket and be replaced with some kind of metal and plastic. So I'm very sensitive to these things right now. Thankfully, at the moment, I think my mind is still in fair shape. So the first thing I have to know is what's expected to be occurring in brain function as people get older, just under normal circumstances. Then I'm going to spend some time getting into some real careful detail about what kinds of things are being complained about. Is it just that you can't remember where you put the remote control, or is it that you can't remember how to use the remote control? Right? Those are two really different kinds of problems. I'm going to take some kind of objective measure of how well your brain works. I'm going to, if I see that there's something is wrong, I'm going to want to start first by looking for things that are sort of easy to fix. A blood test that's out of whack, a medication or something else that might be causing the person's mind to function less well. And then if there is something that looks more like this condition, dementia, I want to see if I can figure out what kind of dementia there is, right? So dementia is a general term, that like automobile is a general term, and then there are subtypes within the category of dementia based on primarily on what the causative mechanism is. It's not all Alzheimer's disease. You can get dementia from having a bunch of strokes. You can get dementia from having a condition that forms a bunch of Lewy bodies in the brain. You can get dementia from having increased fluid buildup inside the cisterns that are in the center of the brain, pressing on the structures of the brain. For example, all of those different types exist. And I'm going to want to see if I can tease out what's the most likely type. And then most importantly, I'm going to want to find the things that I can fix. So this is a cartoon, basically. You can see these are not actual pictures of tissue, but they're a cartoon. And the point of the cartoon is that our brains are shrinking. My brain has been shrinking since I was, about 19 or so. And it continues to shrink. I always talk to the students with me that in the course of the lecture, like in the course of this little time we're together, thousands of brain cells are going to perish. Just as a matter of course, not necessarily because of anything I'm doing, but just, because there is attrition of neurologic cells as we go through time. A lot of that is fortunately surplus brain capacity that you don't use all of the time. So there are structural changes, and if somebody takes a scan of a 20-year-old's brain and a scan of a 70-year-old's brain, you're going to see some degree, most likely, of shrinkage in the 70-year-old's brain. So if you go see a doctor and they take a scan of your brain and there's some shrinkage there, they shouldn't be telling you that you have dementia or diagnosing anything on the basis of the fact that the brain is a little shrunk compared to what they think is a normal brain. And that's important because I do see that happening quite a bit. And this is just another sort of a bunch of graphs, different parts of the brain showing, you see those curves are all downward. Those are just measures of how many cells are in there. You okay? Yeah. Oh, I saw a hand out there. We're good. Okay. So these are different parts of the brain just showing that there's some shrinkage going on. No big surprise. And the top one there, the top set of pictures on the first row are what's called the gray matter. That's where the sort of the processing of information is done. The next row is the white matter. That's like the conduction cables in the center, which manages the information. And on the bottom, you see CSF, which is fluid, which is increasing in amount because basically some of the brain is gone and that space has to be filled, so it gets filled with a little more fluid. That's what we see. And so if you take pictures of young on the top row and old on the bottom row, you see more sort of cauliflower looking on the bottom, right? These are CAT scans. And these are just to show you the pictorial representation of what I've explained, which is our brains are shrinking. Not to worry. Now, the fact that the brain shrinking could be a concern, right? Because you'd think having less brain might be a bad prognostic sign. But it is interesting to see, if we do some specialized testing, that the brain itself works differently as you get older than it did when you were young. This is a good thing. You know, when you think about getting old, one of the things you feel like you have is some wisdom, perhaps, gaining from life experience. Your kids may not always all agree with you 100% on that, but you also process information differently. You've learned ways of sort of packaging things up and routing information and handling it in ways that you didn't used to when you were 20, thank goodness. And so you can see here on the bottom, or in the middle, the yellow ones, the picture is showing different areas of brain activity being stimulated by inputs that are coming into the older patient's brain as compared to a younger patient's brain. So we can measure those things. This is using something called functional MRI. You've heard of MRIs. So functional MRIs are showing the degree of activity of the brain in different parts after you do something, like show a picture or play some sounds or music or a scary sound or whatever else. You can see different areas of the brain light up. And so what these are is they're pictures that people have been shown information of different types, and you can see how the brain processes that using this very special technique, which is a research technique. You wouldn't see it in my office. So now I'm going on to talk a little bit more detail about the diagnosis of dementia. You remember this is a broad category, includes Alzheimer's disease and several other types of neurocognitive impairment. So we changed the technological term that we used to describe this a few years ago. They did this, I think, to upset all of the doctors and all of the record-keeping systems. So instead of saying dementia, now we say major neurocognitive impairment, which is a mouthful. but it's the same thing, you know, when you get to the end of the day. And in order to make this diagnosis, we have to show that the person, number one, has had a decline from their normal level of function. So there are some people who have misfortunes in life and have impaired mental function throughout their life or for a big portion of it based on, you know, conditions or circumstances that have affected that individual. So having that is not the same as having developed dementia. Dementia is something that occurs in somebody who started out up here and is now, you know, dropped a level in terms of their brain. Number one. Second, not explained by other causes. So you can have impaired cognition from a variety of things that have nothing to do with dementia. And so going into politics, for example, is likely to cause some impairment of cognition in my experience. That would be an example of an external influence, right? So that would be another condition. It has to have lasted for 6 months or more, and it must be sufficiently severe to interfere with your normal ability to do stuff. You know, go shopping, call your doctor, get your prescriptions filled, drive your car, pay your bills, pay your taxes, that kind of stuff. When it gets to that point, we know that there's something a little bit more serious going on. And that's kind of our threshold for saying that it's a serious problem. So then if you come to the office and we're trying to figure out if it's more than just a nuisance that you have that you're dealing with, there are a bunch of quizzes that we can do in the office. These are not sophisticated tests, but they give us sort of a clue. And we can do these tests in about 5 or 10 minutes. It's standardized and they've been well validated. There's a whole bunch of them. I'm not going to spend any time telling you in more detail what's in those tests, but some of you may have seen that done in the doctor's office, or they'll have you drawing clocks, ask you a bunch of memory questions and things like that. Or you can go see a psychologist and spend about 3 hours answering the same kind of test, except a lot more questions and a lot more details. Not very expensive, but it gives us a much more precise estimate of what's wrong. What type of problems are we having in this brain? So these are our tests. Oh, go back. Yeah. And then if there's something that is not normal, I'm going to do these blood tests out of the gate, just to make sure that it's not like your sodium is too low or your calcium is too high, or that your B12 is low, thyroid is not working, things of that sort. Occasionally we'll do some additional blood tests based on conditions that we know can affect the person's mind, after taking a little bit of a history to see what the person's been doing with their time. So for example, people used to have fancy hats. I was usually known for wearing hats this time of year. Not really fancy hats, and the hatters who would clean your hat, use mercuric chloride to clean the hats, not knowing at the time that mercury is really bad for your brain. And so then you know the expression mad as a hatter. People who were cleaning hats with mercuric chloride would get mercury on their hands, they would get it into their blood, the mercury would land up in the brain, and pretty soon the brain wasn't working. So we know that there are things out there in the world that can cause the brain to not work, so we want to make sure that's not what's going on with you. So if you tell me that you've been working as a hatter, I'm going to take a different approach. And then most often, but not always, we're going to get some kind of a picture, a CAT scan or an MRI of your brain. And my purpose when I'm doing that is to make sure that you don't have one or two things like a multitude of strokes or a big blood clot that's caused some changes in your brain function. I've seen this in actuality. I've seen a person who got a gigantic subdural hematoma and his mind had really deteriorated rapidly. Once the subdural hematoma was removed, he was right as rain, back functioning normally. So, we have to make sure that kind of thing isn't the source of the trouble. And then there's a condition where fluid can build up inside the cisterns in the center of the brain that can produce some really wacky changes in the way people function. So we'll get scans in many but not all cases, and be careful not to misunderstand. If your brain is a little shrunk on the scans, that's not any basis for diagnosis. So we shouldn't be ordering MRIs and CAT scans to tell somebody if they do or don't have dementia. We would be using MRIs or CAT scans to see if we can figure out why a person is having cognitive problems. That all make sense? Yep. All right. Everybody's still going with me. That's good. So now we'll get into some much more interesting tests that have come into the mix in the last 15 or 20 years. So one of those is called PET scan, and that stands for positron emission tomography, PET. And it is a test where you're going to give the patient a chemical, which is glucose, that's got a label on it, and that label is a radioactive fluorine molecule, which emits radioactivity. And so the glucose is taken up by the brain cells, and so wherever that stuff concentrates is showing that that's active brain. it's taking up glucose. That's a sign that those cells are healthy. And so you'll see more signal from those areas of the brain where there's a lot of fluorine that's been taken up. So on the left side, you see a normal level of uptake of the 18F glucose on the left-hand, most left-hand column, and then the second column there, you don't see as much orange and red. And so orange and red is the computer just enhancing these images to say we're seeing a lot of fluorine there. So that's telling us that second column of pictures is from somebody whose brain is not as metabolically active. And that's concerning, and that suggests there's something wrong. On the right-hand side, past the red line, are pictures that were taken from in a slightly different way. This is also PET imaging, but what we've done is we've given the patient administered a chemical which is designed to bind to a compound called amyloid. So amyloid is commonly found in most people's brains in small quantities. But in people that have developed Alzheimer's disease particularly, there tends to be an excessive amount of amyloid building up as a result of whatever that disease process is. And so on the right-hand two columns, you're seeing where there's more red and yellow. That's bad, because that's activity where the radio tracer has attached itself to amyloid, and you're seeing excess amounts of amyloid. So the third column are normal amounts of amyloid, not very much. And the right-hand column would be somebody that's got quite a lot, probably has Alzheimer's disease. So this is just to introduce you to that imaging technology, because we're going to show that again in a little bit, because I'm going to spend some time talking, particularly toward the end, about amyloid. Good so far? Everybody with me? All right, so now this is a group of pictures, and I have a reason for showing you all these pictures. This group of pictures across the top are people with, on the far left, all the way over on the left-hand side. Let's see if I can do this. Right there, that guy. That's normal. This is a person that's got Parkinson's disease, and Parkinson's disease in its later stages can produce a dementia kind of picture as a result of the process that's involved in Parkinson's. This patient has Lewy body dementia, okay? And you can see this person is starting to show a fair amount of that amyloid stuff showing up on that scan. This person has mild cognitive impairment. That means that you have something that's affecting your ability to process information, but it's not quite at the stage of being dementia yet. So this is somewhere a little beyond senior moments, but not all the way to where you're not able to figure out how to use the remote anymore. Now, I gotta admit, at home, if I'm trying to use the remote, 9 times out of 10, if it's more than just a little bit basic remote use, I have to turn to my wife and say, could you help me with this? Because she's much better at using it. I'm talking now about, thinking the remote is the telephone, or trying to turn on the TV. No, not the TV, but the microwave with the remote, the TV, something like that. So all right, so this is mild cognitive impairment. This is Alzheimer's disease. You can see there's a whole lot of amyloid. This is that frontotemporal dementia, right? So that's the same thing that you might have heard has affected Bruce Willis, that was also the disease that took down the comedian, I'm forgetting his first name, Williams. Robert Williams, yeah, thank you very much. And my brain is okay. And we'll come back in a second. This one's vascular dementia. See, it's not much amyloid. So my point in this is to remind you that there are a whole bunch of different forms of dementia in terms of the mechanism. Not all of them involve the same chemical process. So when we're talking about Alzheimer's disease, we're starting to think of it more and more as a condition that has a lot of connections to amyloid in a pathological protein called tau. And it is a different mechanism from some of these other kinds of dementia, which all of them affect our society. So it's not just Alzheimer's. Okay, there. So now I'm going to talk to you a little bit about the available drugs that we have. So If you watch TV at all, you've seen at least a million ads, right? For one or other of these products here, Nariva, Prevagen, and a bunch of others. You may have heard of ginkgo biloba. These are what are called nootropic agents, and they all share one general similarity, which is they have no beneficial effects that have been ever scientifically demonstrated in enhancing memory. They have all really great marketing plans, but I'm going to tell you, I don't mean any offense. If you're swallowing these things and believing in them strongly, that's your choice, but it's not going to save you, okay? It is just not, there's no evidence of that. Then there's this group of drugs right here. Okay, this group of drugs right here is a group that we give to people who we're pretty sure have dementia. And the first three right there, they have the common property of raising the levels of a neurotransmitter in the brain called acetylcholine, which has been somehow found to be more likely to be affected in diseases like Alzheimer's. We don't know why, but somehow the nerve cells that use acetylcholine as their way of communicating are more susceptible to the condition that produces Alzheimer's disease. And then there's this drug down here, Namenda, which is it works in a different way. It decreases the activity of a neurotransmitter called glutamic acid, which is excitatory. My way of thinking about this is that maybe when the brain cells are tired, they don't like being excited. I'm not sure. But Namenda works differently. It reduces the activity of glutamic acid as a neurotransmitter. The thing about these drugs is they don't cure anything. You know, they don't stop the disease from progressing. They don't keep you from going normal to mild cognitive impairment, from mild cognitive impairment to full-on dementia. But they might make it so you do a little bit better in the course of a given day or over a span of a year or 2 or 3 by taking these drugs compared to people who are getting the placebo, the sugar pill, you know, the not active drug. So that's what we have. That's what we have had for quite a few years. Unfortunately, you know, because we're looking for something that's going to make this all better. We just haven't had anything. So now changing subjects again for a second, because I always get this question usually from the kids of the patients. Yeah. So you might have seen this movie, right? And sometimes the patients, because they're worried about their kids, but mostly saying, well, I have 3 relatives in my family who have this condition, or both of my parents had it, or something like that, and they wonder if they're susceptible and going to get dementia. So is it genetic? And now, this is going to be a little bit of a digression back into your basic neurosciences class in genetics. You all remember that back when? Not everybody went to school to learn science, I know. But I need to teach you just a little bit about genetics for this part of the talk. You guys ready for this? Yeah? Okay, good. All right, so what we learned a bunch of years ago is that all cells, you know, on the planet of every kind, every form of life, cows, grass, mosquitoes, whatever else, they're directed, the cell's work is directed by this group of proteins called DNA. And then that DNA gets copied onto, it's like a carbon copy format, but it's a different chemical called ribonucleic acid, or RNA, which then goes over here to the mitochondria, if I'm able to get the pointer right there, and shows the mitochondria the code to put these different amino acids in a sequence, which then becomes a protein. And our body is full of all kinds of proteins. So the DNA is the code book that determines how we're going to be, how tall we're going to be, what color our eyes are going to be, that kind of stuff. It's translated over to RNA. It's translated into proteins. The proteins are folded. They naturally tend to fold. And these little proteins, which are all through all your body, are the things that are making things work in you. And this is showing you the sequence of the code blocks that make DNA. There's only 4, actually, different types. So all of what makes us is an alphabet with four letters, essentially. And so if you have a mistake, like for example, right here, that's the normal sequence for this particular gene, and there's a mistake down here, and what you end up with is you end up with a mutation that turns this normal hemoglobin into a sickle cell hemoglobin and puts you in a circumstance where all of your little red cells are going to crumple up and become deformed and die when the oxygen level goes down and cause you terrible pain, cause you to have strokes, become, very sick. It's a genetic disease. It's made by one error in one gene on chromosome 6. And so a lot of what goes on in our bodies is dictated by these genes. And so now the question then becomes, is there a gene or a group of genes that might be responsible for causing us to go from being normal cognitively to developing one of these conditions like Alzheimer's disease? So the answer is no. People have looked. We have actually decoded the entirety of the human genome in a project that's been going on now for about 20 years. But people have taken human cells and human DNA and broken down each gene into those 4-letter blocks to see what the gene is made of. So there's this whole long string of letters, essentially, that represents each gene. So we know what these genes are, we know where they are on the chromosomes. We have 23 pairs of chromosomes, 46 total, right? And we know where these genes are on the chromosomes. We don't know what most of them do, but we know where they are and what they look like. So then what you can do is you can put that information into a computer. We can take all of these gene samples from patients, some who have normal brains, some who have dementia, and look to see if there is any association between those individual genes that we know exist and the development of these conditions. And what you have on the bottom here is you have all of those genes down to number 22. We didn't have the sex chromosomes on this particular study, but all of the somatic chromosomes right there. And then each of these signals above this little dotted line is that there is some association with dementia. So what you can see is there's just a whole bunch of genes. No one gene or no one cluster of genes, but a whole bunch of genes that have some possible association with the development of dementia, which is a way of simply saying that we're not going to find a single answer. Like, remember, for the sickle cell disease, there's one mistake on chromosome 6, and that's it. You have sickle cell disease if you have that one error. You're still with me? Who's laughing? Yep. All right. So now, people have tried to improve on our inability to predict by producing a score, basically looking at all the genes that you got. If you have one of those, remember there were a whole bunch that had above normal levels of connection to Alzheimer's disease in the previous slide. So if we take all of those 83 or something genes that might have some association with dementia, we can produce a risk score for you. So whether I would want to have this risk score done for me, I'm not sure, because it would make me not sleep well at night. But then I can predict that if I had a high score on the combination of all of those genes, it's more likely that I'm going to wind up getting some form of dementia later. So let's go back to see if there are any individual genes that might be causing an actual case of dementia. So there are a couple of locations where genes are more directly connected to the diagnosis of dementia. there's a gene locus called apolipoprotein E. It's an important chemical in cell membranes. And there are basically 3 types of apolipoprotein E. There's E2, E3, E4. And what we know is that if you have 2 copies, meaning both of the chromosomes that carry that gene, there's a pair of chromosomes that carry that gene, if both of them have apolipoprotein E4 type, then your chances of developing Alzheimer's disease are 19 times higher than would be the case for the average person, or let's say a person that has 2 copies of E2 or E3. So apolipoprotein E4, especially if you have 2 copies of it, that's not good. It makes your chances significantly higher of developing dementia. And then there are families that have these mutations in these 2 genes called presenilin 1 and presenilin 2 that have a very high likelihood of progressing to early dementia. So this is me apologetically saying that even though almost all cases of dementia are not genetic, there are a few families that actually have a genetic predisposition to getting dementia. Fortunately, these are rare, super rare, 1 in hundreds of thousands, or 1 in several million to have these genetic conditions that predispose to dementia. So almost none of us are going to be affected by this. In almost every case of Alzheimer's disease, even in families where 3 or 4 people have Alzheimer's disease, it's not going to be primarily a genetic disorder. It's just going to be what we call sporadic. Is everybody clear on that? Any questions? Okay, so good. All right. Moving on. So now I'm going to tell you a little bit more about this amyloid story. And towards the end, I'm going to tell you why this amyloid has become such a subject recently, which is, that there are drugs that we've seen come onto the market that can help us with this amyloid problem. So the story with amyloid is that in patients that build up amyloid, so this is the amyloid buildup right there, in patients that are predisposed to building up amyloid, and they've not yet been diagnosed. So down here, this is preclinical. All of this time, their function is, biological function, your brain function is normal. Now in here, this is mild cognitive impairment, and up here, this is dementia. So first, there's amyloid. And that can precede the development of clinical dementia by 15 years. So it could be like lurking back there in any of us, right? I might have some there. I don't even want to think about it. I certainly wouldn't want to be scanned for it. Then we start to see evidence, if we're following people over time, we can see evidence that the brain cells are not talking to each other as well. We can start to see this protein tau showing up in the cerebrospinal fluid. Now we're starting to see structural changes in the brain. It's shrinking faster than normal. And now this purple line is, we're starting to see changes in cognition. So this is all going on in the background before somebody shows up in the doctor's office that's really having trouble doing things cognitively. So there is a general belief that there's, in some patients particularly with classic Alzheimer's disease, that somehow this protein accumulation, tau and beta amyloid, is contributing to the disease state that we all know as Alzheimer's disease. All right, and I'm jumping back to the genes again for just a quick second. So these very few families that have this unfortunate genetic problem of a mutation in the presenilin gene, that gene makes this protein. Okay? And all of these little red circles are places where there's been a detected mutation at some point in these individual families. So it's not all just in one exact spot, but if you get a dysfunctional presenilin gene and your body's making this type of abnormal protein, it's bad. So here you are 35, and you have asymptomatic pre-MCI, means we can just detect some evidence that your brain is having some problems. Here you are at 38, and for 6 years you're having symptomatic pre-MCI, means that you're actually having some trouble with your daily activities. For 5 years or so, when you're 45, you're having problems with mild cognitive impairment, and here you are at 49, and you have 10 years of dementia before you die. So if you have one of these presentinel gene mutations, it's a really bad thing. The reason I'm bringing this up is because these patients will show evidence of building up a lot of amyloid. And so there's been a lot of study conducted on these families just to see what's going on in the brains. And there are multiple slides here just to kind of highlight the bad effects of having the presentinel gene. So these guys here with the red boxes are symptomatic carriers, means they've got symptoms of the condition. These are people in gray blocks who are non-carriers, so their brains are fine on imaging, and their brains are working okay in real life. But if you get the presentinel gene, you end up building up a lot of amyloid, basically, and start to lose it at a relatively early age. It's a bad thing. Yeah. So going back and forth just a little bit, so I've loaded your brain with this notion that there's amyloid as a problem. We have lots of lines of evidence now to suggest various different mechanisms in addition to amyloid, because amyloid isn't the only thing. Remember I showed you at the very beginning there's Lewy body dementia and there's frontotemporal dementia, and those people don't have a lot of amyloid. There's multi-infarct dementia. There's a bunch of things that can happen to your brain that don't involve amyloid. So these are all different theories about what else could be going on. High cholesterol, this is the amyloid presentinel thing. Then there are just a whole bunch of different mechanisms that might be contributing to the development of dementia, still being explored, scientifically not agreed on yet. And this is another picture of the doctors and the scientists putting their heads together and trying to figure out, again, if there's any way of tracing this back to one or more genes, really problematic, and the answer is basically no. There's evidence that there are some risk factors that are genetic, but not any one mechanism that's coming out. All right, so that's the basic understanding of dementia and its underpinnings, right? Of course, none of us really want to have that ever if we can avoid it, right? So, I mean, I'm really hoping I don't at any point. My mom had it, so I saw what that looked like. It was not good. So what can we do? And there's been a lot of discussion of this. One thing, of course, is sort of healthy lifestyle types of things that make sense. How many of you all here are sort of Mediterranean diet people? Hands up. Mediterranean diet. Listen, halfway hands up. All right. How many fans of olive oil as a way of staying healthy? Just a small group. Not that many Mediterraneans and not that many olive oil drinkers, huh? So we'll talk a little bit more about that in just a second, because there is some potential benefit diet-wise in terms of protecting your brain. Obviously, we know that there are some things that are generally bad for your health. So getting atherosclerosis, hardening of the arteries is not good for your brain. So controlling blood pressure, controlling cholesterol, probably helpful. Exercise, that sort of stimulating blood flow and does other good things for you. Staying socially engaged and mentally active. So, you see people now trying to do exercises for their brain to see if they can keep their brain from developing dementia. Does that really work? Probably not. But it does help to stay engaged in stuff, you know. So engaged in life, reading books, talking with people about things that are, you know, complicated subjects, solving problems for yourself, for others, doing your taxes without the assistance of a computer. Those kinds of things for sure will help your brain. They may not help your bottom line, but they'll probably help your brain. Next thing is to avoid anticholinergic drugs. So there are a lot of drugs out there in the world that have chemical property of being anticholinergic, meaning blocking transmission by acetylcholine. And so any drug that you buy in the drugstore that has like a PM on it, if you look at that label, it's going to say diphenhydramine on there, which is the same as Benadryl. Those of you who maybe have taken those drugs know that they help you become more sleepy, and you may take them for that reason. If you're an older male, you may also know that they tend to make it harder to pass your urine. And whichever side of the aisle you are, those drugs make it harder to move your bowels. They dry you out, and they affect several autonomic functions in your nervous system. Those drugs may, if you take enough of them over the span of a lifetime, may contribute to a predisposition for developing Alzheimer's disease. We don't know for sure, but people who've been in medicine have now been arguing about whether we can do stuff to keep people from getting dementia for quite a while. So the bottom line is take care of your vessels, exercise, stay socially engaged, and probably you can gain some benefit from the Mediterranean diet. This is just a sort of a summary slide that touches on all of the various things that are probably bad for you that you shouldn't do, or that you could do less of. Smoking cigarettes, not so good. Physical inactivity, social isolation, not so good. So those things are favorable. Let me just tell you about the olive oil thing before I leave that alone. So the Mediterranean diet was studied quite extensively in Europe. They had a study of thousands of people, mostly in Spain, who were middle-aged and older, predisposed a little bit towards hardening of the arteries, and they put them in a study where there were 3 branches. There was the branch where you just got advice, and they got a nice brochure that said basically don't eat all that rich fatty food, eat this other healthy food, and exercise. And then there was a branch where they got the brochure and instructions to eat a bowl of mixed nuts every day and to not eat as much animal fats and butter and things. So they were substituting all the oils that were in the mixed nuts for the oils that were in their regular fatty diet. And then branch 3 was people who had olive oil in place of animal fats and butter. And they had to drink a fair amount of olive oil. So those of you who are olive oil drinkers, let's see how dedicated olive oil drinkers you are. Who drinks a quart of olive oil a month? Anybody? Maybe one half-hearted hand. That might be a quart. So anyway, these guys had a quart of olive oil a week and 4 ounces of olive oil a day. A lot of olive oil. That's a heck of a lot of olive oil. Yeah, and I have historically run a conference here that's been sort of sabotaged by COVID year over year. We had, several hundred people used to come to this conference. It's sort of on hold right now while I'm looking for sponsors to help me bring it back up. But we have all these physicians from all over Central Virginia and down into North Carolina and other places. One of my friends who used to come and speak at the conference, he has diabetes. He's a world famous biological scientist related to aging. He's a gerontologist and an endocrinologist. So John was at the conference when I gave a talk on nutritional strategies to stay healthy. And I asked him, I did this olive oil thing, in this crowd of doctors, 2/3 put their hands up there drinking olive oil to stay healthy. Well, keep your hand up if you're drinking, a quart of olive oil a month. The hand's coming down. Keep your hands up if you're doing a quart a week. So John's hand stayed up. John, how much olive oil are you drinking? 2 quarts a week. So he said, I slathered on everything. The point of that is that you're substituting polyunsaturated fatty acids for saturated fatty acids in your diet. And the reason for telling you this story is in this randomized controlled clinical trial where they had thousands of these guys in Spain who were at some risk of developing hardening of the arteries and stuff. They also did a sub-study where they looked to see how many of them got dementia, and it was less. And so, there is some scientific evidence that says maybe this kind of strategy can work, but you have to really go at it. And this is just an analysis of cognitive function tests from that study, where they looked to see how well people could do who were on the olive oil branch, and they did better. I'm just saying. So I'm not at a quart a week personally, but I'm probably halfway there, and I'm going to continue to press the agenda, right? I think it makes a fair amount of sense. You have to sort of drop some of the things that you otherwise love to eat that taste really good. And I'm not saying that olive oil tastes bad. Sometimes when I give this talk, I bring like a big jug of olive oil and have a couple swigs while I'm giving the talk. So, consume more PUFAs, polyunsaturated fatty acids. That's a strategy. And this is a kind of complicated study, but it basically is to let you know that taking those anticholinergic drugs, whether they're prescribed by people like me to help you control your bladder, to help you control Parkinson's disease, to treat depression, or if they're consumed by you on your own, going and buying Tylenol PM, or whatever else it is that says PM with lots of Benadryl in it, anticholinergic drugs may predispose towards later developing dementia. So maybe a bad idea. Leave those things go if you can. There. So this is just to remind me to tell you that there's like millions of anticholinergic drugs out there. Some of them are really anticholinergic, like Benadryl, not very good for you. And then some others that you wouldn't think are anticholinergic, like ciprofloxacin that you may have had for infections and things like that. And of course, you wouldn't be taking ciprofloxacin every day of the year for years and years. And it's not highly anticholinergic, but this is just to say there's a ton of anticholinergic drugs in our world, and they may not be best for our brain. Okay. One more thing you might do to help. You might concentrate on getting better sleep at night, because there's a theory that somehow while you're sleeping at night, there's this substructure within the brain called the lymphatic system, which is basically there are cells called glial cells that are the support structures around the nerve cells that do all the work in the brain. And those glial cells during the nighttime, maybe one of the things that they're doing is just basically gathering the garbage, might be including the amyloid proteins and the tau proteins, and taking that stuff out and dumping it from your brain. So getting more sleep at night may be helpful to your brain health. All right, so now I told you at the beginning that we were going to wrap up at the end by talking about new drugs that have come onto the market. So all kinds of people have been doing research to unpack why we're getting dementia and Alzheimer's disease and try to find ways to fix it so you won't have sort of brain rot as you get into your golden years, right? So last was a year and a half ago, maybe 2, a drug called Aduhelm was marketed. It was marketed prematurely because it was the first drug that was found to have been shown to essentially extract amyloid from people's brains. The only problem with Aduhelm was that it also caused quite a lot of complications in terms of brain swelling and brain hemorrhages, 40%. So that's a pretty high price to pay. And so the FDA approved it, but they regretted approving it, and the sensible marketplace rejected it. Nobody really wanted to take that stuff, too poisonous. So now, this year, in January, a randomized randomized controlled clinical trial. That's where you give people the active drug versus something that's inactive, placebo, sugar pill, whatever. Came out, was published with thousands and thousands of patients who were given, like the son of Aduhelm, which was this new drug called lecanemab. It's an antibody that goes into your body through the venous system. You take it as an IV infusion. It gets to the brain and it is able to bind onto amyloid, and then over time, that amyloid becomes removed from the brain. And so you can get, in the span of 18 months, about a 60 or 70% reduction in brain amyloid by infusing this stuff every couple of weeks. It's expensive, $25,000 for a course of treatment. You have to go every 2 weeks and get the infusion. And it's only approved for people that have very early and pretty much proven Alzheimer's disease, because it has about a 20% chance of causing some of that same other thing with brain swelling and in some people brain hemorrhages. It doesn't happen all the time, but it's enough to be a worry. So in addition to being expensive and having to go for infusions every 2 weeks, there is that potential risk. So it's only approved to be given to people that we're fairly sure that they have Alzheimer's disease. So we've shown that they have amyloid excess in the brain. And we've shown that they have enough cognitive impairment that we're going to say they have early Alzheimer's disease. So they still pretty much seem normal to you in conversation, but they're making mistakes more than they should, more than they used to, right? So you have to do some kind of test. So in this case, you either have to do those specialized PET scans where you give that tracer that makes the bad stuff light up in the brain, or you have to take some cerebral spinal fluid and send it to a lab and show that there's an elevated level of amyloid and or tau protein in there, so that we know that they have an amyloid problem, because this drug is an amyloid remover, right? So then you can, if you have those two things, very early dementia and an excess of amyloid, you can go and get this stuff infused. It costs some money. You have to have a bunch of MRIs to make sure your brain is not swelling and bleeding. And then see if your brains are more functional. In this clinical trial that was published this January, there was a slight improvement in cognitive function. That's the most encouraging part. Not only was there a very impressive reduction of the amyloid, there was also a little bit of evidence that the person was improved cognitively. And now there's the cousin of the vote. Oh, that was quick. That's the button I wasn't supposed to push. So these are the slides that show the improvements that occur. So this is the amyloid being reduced right there. This is the drug being, and this is the placebo. So lots less amyloid. That's 70% right there after 18 months. And this is the cognitive function. So this is not dropping off as much in the people that are taking this drug compared to the people that are taking the sugar, the placebo. So looks actually somewhat encouraging, right? Amyloid less, brain better. Oops. Now I'm going to start having to go get checked. And now there's little brother of lecanemab, which is donanemab. Pretty much the same story, again, showing less amyloid and cognitive improvement, drug versus placebo. And over time, and donanemab, which was published in May in the Journal of the American Medical Association, looks like it might be even just a smidge better than lecanemab. So we're going to start seeing more and more people taking interest in this, willing to take the risk of some brain swelling, willing to pay the economic price, willing to get infusions every 2 weeks or 4 weeks, donanemab's every 4 weeks, because they really want to do everything they possibly can to not have a degenerative course of Alzheimer's disease led into their life. And so, yes, please.
What is insurance coverage like so far with these drugs?
So lecanemab is FDA approved and Medicare is working on it. Yes, Medicare is working on it. The next thing they're gonna have to work on is those Amyvid PET scans, which you can't get right now, and they cost 10 grand. So basically, for most patients that are gonna be considered for this form of treatment, they're gonna need to have a lumbar puncture and have some of their cerebrospinal fluid sent up to the Mayo Clinic in order to prove that there's amyloid. So that's what that looks like right now for those who would be qualified, who have the very early stages of Alzheimer's and definitely have some amyloid problem, right? It doesn't have anything to do with Lewy body disease, frontotemporal dementia, or any of that other stuff that's out there. But for this, this Alzheimer's biopathology, there's something. And enough slides of graphics. So this is a graphic to show all of the various drugs that are in various stages of testing to treat Alzheimer's disease. Not all based on amyloid extraction. There are several different mechanisms that are proposed to reduce the burden of the illness on your brain. So that's to say, there's quite a bit of work being done and things in the pipe. So maybe that's a little hopeful. It's going to be a little while before, it takes a while for this all to translate into real life where we live. Yeah, and so now I'm wrapping up. And so this is a reminder, this was from your own website, right? So this is the team page of Cedarfield showing progression towards goal in terms of your fundraising. Pretty good. The Alzheimer's walk coming up pretty soon. I'm not going to be walking. I used to go, and I did it a bunch of years. I took my son with me. We went walking. He still got one of the t-shirts. He likes to wear it. Wrapping this up, it's a very complicated problem, this dementia thing. I think you probably understand now more about how complicated it is, having listened to me prattle on here for the last little while. There is some progress being made, but we're a ways yet from where we need to be. So this would be one of the reasons to contribute, right, to things like this effort that's taking place at the Alzheimer's Walk. And the Alzheimer's Association in Richmond is a very strong chapter. So, supporting them is a great thing to do. I see, a couple of you have your purple shirts on. I have my purple shirt and purple tie, just as a reminder. So I want to encourage you guys, if you're able to walk, get out and walk. If you're not able to walk, get out there and write a check. And there's my information, my email address, and a pretty picture to close up and let you know that I'd be glad to answer any questions.
On that, on the drugs that you were talking about, that possibly coming down the pipe, what age person, or is that just a personal decision?
You talking about these drugs that have been approved for use, or the ones that are being explored scientifically?
The ones that are being explored.
So the way that drug development goes is the people that are trying to put a drug out there for consumption in the marketplace, to be able to be prescribed, you have to do phase 1, phase 2, and phase 3 trials. You have to figure out, first of all, does it do something? Does it do what you want it to do? Then you have to find out if it's poisonous or not too poisonous. And then you finally have to show that given in the recommended therapeutic doses, it produces the benefit that you want to see by having now given it to a bunch of people and compared the drug with the placebo. And so the way these studies go, they take people who are selected because they either have a condition or they're likely to get a condition to test them. And so it's not restricted to people of advanced age. Many times these drugs are being tested in people who are sort of pre-advanced age. You wouldn't probably want to give these drugs so much to 20-year-olds, other than there are a lot of 20-year-olds who have questionable thought processes. Our brains are not really mature when we're 20, so you make all kinds of bad decisions. I'm sure many of us did in our 20s. But the point is that, you want to selectively give the drugs to a target population that you think are likely to benefit. Yeah.
Looking back, say 50 years, has the number of cases of dementia increased? Something I didn't hear about when I was young. Maybe that's the reason I didn't hear about it. And then projecting ahead, are there any projections of how much the disease, especially with baby boomers getting in for being diagnosed, how are we prepared or not prepared for that increase?
Great questions. So those sort of population health questions, and those are terrific questions. So I'm going to say that this condition is not a new condition. It's been around. What's new is that the information is much more available now. You get the news on a pretty much continuous basis, right? One way or another. So you're hearing about these things more, and doctors are being better educated about these conditions, and they are making a diagnosis sooner and more accurately. So a certain portion of the apparent increase in dementia is that we've recognized what it is and we're calling it by its name, and so you're sort of more aware of it in that respect. The other thing, of course, that's happening is that our population is aging, in the sense that we've got this baby boom thing going on, right? A whole bunch more people were born in the 25 years after 1946, right on the tail end of World War II. In a big bulge, that group of people are moving up in our society, so proportionately our society is older. And this is, for sure, a condition that is much more common as you get into advanced age. So past age 85, the chances of having dementia at some point in life, probably a third. If you live to that age, your chances of developing dementia are fairly high. But it's not 100%. So you can be trucking along, doing okay cognitively when you're 100, 150, 120, as long as you've been fortunate in terms of your biological health and your lifestyle, and probably to some extent your genes. So it's not a guarantee, but it is an age-associated disorder, which we don't really know exactly why that is. Maybe it takes a while for this stuff to build up. We do know that many longitudinal studies of people at risk for dementia have had preclinical amyloid buildup in their brains, that for those who eventually got Alzheimer's disease, that preceded the diagnosis by about 15 years. So some of it is living longer, and some of it is baby boomers in the world. My son, who's 13, he calls me a boomer when he wants to say that I'm not in touch with contemporary thinking. And apparently it is a term in middle school. That's boomer thinking, Dad. So
We call it old fogies.
huh?
We call it old.
Oh yeah, well, focus, right. So I think it's a combination of more information out there, an aging population, better diagnostics, better understanding of the disease states, and changes in terminology, all of the above. Yes, sir.
Where would I find the statistical data that relates the age of diagnosis to the age of death for the different types of dementia?
Oh my gosh. Well, so I go generally to the National Library of Medicine when I'm trying to look that kind of stuff up, because there are just bunches and bunches of papers. Generally speaking, on average, dementia from the time of clinical diagnosis, that's the point where somebody, you or your family member or somebody, dragged you into my office and said, things aren't right here, see what you can figure out. And then I do all the things that we talked about at the beginning, and I say, I'm sorry to tell you, you have some form of dementia. From that point in time, on average, it's about 10 years to where you're basically reduced to inability to do much of anything. Feed yourself, talk, walk, on average. Some forms of dementia, like if you have that misfortune of having a presenile mutation, much more rapidly progressive. Some forms of Lewy body dementia tend to be faster. Certain other kinds of conditions faster. Some others a little bit more indolent and slower. Some people go along with dementia for 15 years, 20 years even maybe before they start to become end stage. Average about 10.
So the short answer is about 10 years until you need until you are, I don't know, fully demented.
Right, end stage,
then-
I'd say profoundly demented. That means you really can't do much of anything.
And
You
then
can't
4 or
remember
5 years after that, where you would need care.
No, you're already in care at that point. When 10 years down, you're in a facility or receiving care, and you're maybe going into hospice. So the course of dementia, if you wanted to sort of graph it out, there's 3 or 4 years, on average, 3 or 4 years where it's pretty mild and it's just, people are aware something's not right. There's probably another 3 or 4 or 5 years where you're starting to really have significant problems and you need supervision, you need care. And then there's probably 2-3 years at the end where you're really in terrible shape. And in some cases, going to be institutionalized. In some cases, you have a supportive family where you can afford care in the community. But it's sort of like that. That's sort of the time course of it. It's a terrible disease. My mom, had it. She was a brilliant doctor in her day, but it was not a pretty picture at the end.
Any other questions? Dr. Boling, thank you for your time. Thank you for your information.
Well, absolutely glad to be here.
So let's go back to a lot of things in the pipeline.
Things are in the pipeline.
Continue your support for Alzheimer's Association.
Absolutely. More PUFAs and less anticholinergic drugs.
Thank you.